From Longwood in Boston, this is The Pulse, a podcast that dives deep into biotech stories and features biotech leaders. I'm one of your hosts, Yosef Baidi.
I'm your other host, Fang.
The podcast is brought to you by the Harvard Biotech Club, a student-led, Harvard-affiliated biotech organization on a mission to bridge the gap between academia and industry.
Today's guest is Dr. George Church, a genetics professor at Harvard Medical School and the Wyss Institute. George is a pioneer in synthetic biology and personalized genomics. For most of our audience, his achievements need no introduction. In 2017, Time magazine named him as one of the 100 most influential people in the world. In addition to being a scientist, George is also a prolific serial entrepreneur. He has co-founded over 50 biotech startups through his lab in Harvard. George serves as a faculty advisor for the Biotech Club. We couldn't be more excited to have him as our guest to talk more about his prolific career and beyond. Welcome to the podcast, George.
Thank you. Pleasure to be talking to you now.
Can you tell us a bit about your upbringing and what led you to be interested in science early in your life?
So my upbringing didn't really include science much. That I didn't know anybody that was a scientist or an engineer until I was, until I went away to school in Massachusetts. In Florida, the education was weak in science. Didn't even have a science section until 7th grade. But I had a lot of curiosity about it, and so I would, both in the natural and in the unnatural, which is more engineering. So I got exposed to the idea of computers, although I didn't have any access to them. So I tried to build them. It was very pathetic and simple, but it got me thinking about it. And then in the natural, I would study metamorphosis at a very young age, trying to figure out what it was. And I was mildly dyslexic, so I had to kind of figure out what it was in the mainly depending on pictures. So with dragonfly metamorphosis in particular. Yeah, so that was fun. And that got me hooked. That was the hook. And then the problem was figuring out what I was supposed to focus on, and I still haven't figured that out. I'm focusing on everything.
Yeah, that's true. Your lab is like a lot of varied interest in everything. So I guess you got interested in science at that point, and then what specifically got you interested in biomedical research and led you to pursue a PhD?
Yeah. So right. So I was looking for some field that did incorporate multiple brands of science. So, and it turned out that first one I got that really hooked me further was crystallography. So that required, math, Fourier transforms, computer science, biology, chemistry, physics, and medicine. And it was at the Duke Medical Center that I started doing this. And that crystallography was on a nucleic acid transfer RNA, and that led me to typing in all the DNA and RNA sequences of the time, and that led me to Wally Gilbert's lab at Harvard, where it looked like he was really the clear leader in pioneering studies of nucleic acids. In particular, it looked like he was on a path to making a plausible DNA sequencing method. So that I enrolled in I applied and got into the PhD program at Harvard, even though I had flunked out of one PhD program already. But they took me. That was good. I was glad.
Yeah. So you mentioned being interested in the kind of development of genome sequencing methods. So 1980s, you and Walter were instrumental in developing these first genome sequencing methods and publishing it. Could you tell us a little bit more about the backstory leading to the development of these methods? And, this is a very important time because the Human Genome Project started a few years later, or was initiated.
Yeah. So, so Wally was one of the key players in recombinant DNA of cloning things, and as part of my rotations when I was just a first-year graduate student, I rotated through 4 labs, all of which contributed in one way or another to my thesis. Mm-hmm. But in Wally's lab, we sequenced a little bit of of the first recombinant DNA plasmid that was now used in recombinant DNA from then until the present. And that was everything I hoped it would be, because I spent the summer kind of speculating on how one could multiplex DNA sequencing. In other words, doing lots of sequences together. And of course, my first attempt at it was miserable failure. The student that was teaching me to sequence said, "Why don't you just learn how to sequence first, and then you can innovate," basically. And I did that. But then for most of my thesis, I spent developing a genomic sequencing method, and it really was the perfect environment to do it in. And it did. About the time I published that, there was a Department of Energy meeting that where they wanted to figure out mutation rates. That was their congressional mandate, but they couldn't. And we offered that, well, maybe before you do that, you need to have the baseline, which is the sequence of at least one human genome. And so that was the start of the Genome Project in 1984.
It's really cool to hear about that now that we know genome sequencing methods are much more kind of accessible and cheaper, and hearing kind of the initial steps to show the proof of concept, since it needed to be at a much larger scale for the actual project.
Yeah.
So was PCR invented? And PCR, the PCR.
PCR was invented around that time by Carey Mullis. My method didn't use any kind of amplification, didn't involve recombinant DNA of the molecules we were sequencing. It involved recombinant DNA setting everything up, but not. So we would take the entire genome and immobilize it on a solid surface, very similar to what we do today, and do rounds of probing and imaging. So you take the surface, you'd image it, probe it again, wash away the excess, and this was the basis, I think, of next-gen sequencing. 2 decades later.
Yeah. After finishing your PhD, you worked briefly at Biogen and did a short postdoc with Gail Martin at UCSF before returning to Harvard to start your own lab as a professor. Could you tell us more about how these different stages of your career played out as you were figuring out what you wanted to do long term?
Right. So, yeah, that was a very rapid set of changes. Basically, I went from a PhD to Biogen. 6 months later, I was at UCSF working on stem cells. And then a little over a year later, I was applying to Harvard. And the way it worked is, at the end of my PhD, which, Wally and I had this new genomic sequencing method, which we were applying not just to sequencing, but for footprinting and methylation, which are epigenetics. And we were applying it to the immunoglobulin system, which was fresh and new, and it was the first tissue-specific enhancer. Worked with Susuma Tanagawa's lab on that as well, Anna Frusi. And that, I wanted to include lots of different stages in development of the immune system, and the earliest stage were pre-B cells, and even before that I wanted to have pluripotent stem cells. And Gail was really one of the few people in the world who had those, and so I asked her for them. She very nicely sent them to me, and I said, "Oh, this is so cool. I have to apply to a postdoc in her lab to learn more about stem cells," because at that point I was just kind of a molecular biology jockey doing molecular immunology, not real immunology. And so I went and learned, and I pitched to her doing homologous recombination in stem cells. Unfortunately, we got scooped by her mentor, but that was fine. And and then I went. I had followed my girlfriend, now, 48-year relationship wife. We're grandparents now, but anyway, I followed her to the West Coast and back really quickly, and was very fortunate that once again Harvard has saved my bacon basically 3 times. Once was picking me up after flying out. Once was allowing me to come in with very little postdoctoral publications, very few, and then again at tenure, they was very nice to me and giving me tenure even though I had, again, very few publications. and not really recognizable on subjects that people usually wrote about at Harvard. So, cool.
And then we want to go a little bit back more into like genome sequencing. So
Yeah.
I know a big thing that you're interested in is making genome sequencing more accessible, as well as making sure people have access to
Yes.
this data.
Right.
So in 2005, you started the Personal Genome Project to help with sharing genome data.
Yes.
So could you talk a little bit about the impact this initiative has had?
Right. So part of this, so there's been kind of two themes that apply to all the different technologies I've worked on, almost all of them, is that one is anticipating unintended consequences and what you can do about mitigating those before you get, too addicted to the new technology. And so we've done that for synthetic DNA and for gene drives and in this case for personal genomics. And we anticipated the dynamics between researchers who want to promise they want to promise privacy and confidentiality, but they also want to publish information about you, and even if the various things that were tried, I felt were not going to work long term, and so I published a paper on it and started a project. We had at that point a center of excellence to develop next-gen sequencing, center of excellence in genomic science, and in that we had a section on ethics, ethical, legal, and social implications, ELSI, and in that we proposed that we would get consent specifically for sharing without guaranteeing privacy, but instead guaranteeing public, which I felt was more realistic, because there were already many cases where people had left a laptop in the subway, or something had been hacked. Millions of records had been, of even medical records, not just genomic records, had been made public in a way that was not intended. So we wanted to have very clear consenting, and we also wanted to have kind of a test bed for new technologies. And that project is now 2 decades old. We're celebrating the anniversary. So
It's really cool. Yeah, I think it's a good balance of accessible openness, but also confidentiality as well, which is a big topic with people's data, especially sequencing data.
Mm-hmm.
And then we kind of want to talk a little bit about the philosophy of doing science. So you're on the Night Science podcast. You discussed the philosophy of picking ideas to work on.
Yeah.
And you talked about the benefit of working on a lot of ideas and how you can change the world as long as you don't care who gets credit. Could you elaborate on how this has kind of guided you as like a trainee and now as a PI?
Yeah. So, I think occasionally you can get credit and you shouldn't run away from it, but it's amazing how much more you can do if you don't care about awards and money. And sometimes the awards and money and the credit will chase you, and that's fine. That's a healthier situation. But yeah, picking projects is very non-obvious, and it's and you're kind of caught between a rock and a hard place, which is that if you pick something that's too innovative, it's not good to be. Sometimes it sounds very romantic to be way ahead of
Mm-hmm.
he or she was way ahead of their time. It's actually very unpleasant to be ahead of your time, because you're not going to get money. You're not going to get, people are going to think you're nuts. You get no respect, as Rodney Dangerfield would say. So you want to hit it just a little bit ahead, if you can. You want to. When I was starting graduate school, you really had to choose whether you were working on something that was very basic and kind of philosophically interesting and divorced from the world, or pick something that was technological but not necessarily applied to a real world problem, or you could work on a real world problem that you get labeled as being clinical or being industrial, and those labels, basic, technological, industrial, stuck to you, and it was hard to get out. But now, or during my career, since being close to and sometimes often contributing to the technology revolutions, we could see that you could actually do all three, and that became kind of a litmus test for a new project. Is if it hit all three, then there's probably something cool going on there. Social impact, and maybe factors of 10 or factors of 100 in technology, not just factor of 1.1 improvement. And then something that was kind of amusing and basic science, curiosity driven. If you can do all three of those at once, that's just amazing. And we've kind of gotten used to insisting on it.
That's good too. And I guess like probably being a lab where people work together as a team, it kind of promotes that. If you're not going to get the most credit on it, but like you help someone get like a first author publication, it's like great for everyone involved as well.
Yeah. So I do try to get credit for other people, but I just think it's helpful if you have an attitude. Another attitude that took me a while to develop was waste. I got kind of obsessive about watching other people waste money, and I eventually said, no, this is kind of like the leaves in the fall. They make nice colors and they fall to the ground. It's not littering. It's not waste. It's just nature, and it's and we actually have enough abundance in nature. I mean, sometimes it doesn't feel like it with 8 billion people on the planet, but we have enough that we're going to waste a lot, and it's just do your bit to not waste your moments and your money.
Yeah, that's a good point. There will be some waste, but minimalize it.
Yeah.
So oftentimes, when a researcher find outlier in their data, they might ignore them, or while you mentioned in another podcast you like to embrace the outlier, could you tell us about this philosophy? Has it impacted the way you did science or how you advise your trainee?
Yes. So that is something that I emphasize especially in my computational biology courses that I teach. Embrace the outlier. And if you think of life or your experiment as a scatter plot, where you got two axes and you got a nice cluster of points along the diagonal, and then you'll have one way up at the top or off to the side, and you'll say, what's that? And some people will say, oh, that's a statistical error. We're gonna use proper statistical methods to eliminate it, ideally in an ethical and forthright way. But I say, no, that's one of two things. It's either a problem that you might run into again. It's could be some systematic error in your technology or your technique, or it could be a discovery. Either way, you want to study it rather than just use proper statistics to get rid of it. So anyway, and that's only one way to make discoveries. I mean, there are plenty of other ways, but I think it's a good way. It's looking for what's going on, and also it applies to picking people. Science is a lot about picking people, the right people for the right task, and I think in science, at least as much as in other fields, probably more than most, you want people that are neuroatypical, that are thinking differently. And it doesn't really matter on what axis they're. I mean, they don't all have to be. It can be almost anything that causes them to be, ostracized as children, because and so that's again embracing your outliers. Is the outliers in society can often think about things and are willing to take a risk because they didn't spend their childhood conforming and fitting in and even leading social networks. They had to do other things, and so they were, in a certain sense, developed a set of bravery that Nietzsche might say, what doesn't kill you makes you stronger. So that's another kind of outlier.
That's really cool. Yeah, scientific outliers, but also people and personnel as outliers as well, and the value of that.
Yeah.
So even as a PI, we know you still occasionally have time to do experiments in the lab or even do computational work. Yeah. So what's the motivation to continue being active and doing research when you have all these other obligations of being a PI?
Well, there's a few answers. I mean, one is sometimes there's just something that I or somebody feels that I can do better than training people. In other words, it's easier to just do it. My main job is training, but if there's somebody now. Also, there's sometimes the task will be so risky, I think it's actually not good for the career of the students and postdocs. And so I figure my career is pretty stable. So for example, when I encoded my book into DNA, I felt that was going to be relatively easy, but also could be embarrassing, or something like that. And the same thing went for, and then other times it'd be like just obsessed with an idea, and I don't want to have to convince anybody. I don't want to convince granting agencies or students. Some of my favorite ideas took 5 years to convince anybody, and it's a fairly big lab to choose from. I couldn't convince anybody to do, for example, next-gen sequencing for 5 years until Rob Mitra showed up as a second-hand student at MIT, where his professor had left MIT, and he was, I guess, partially desperate, but I think he also had a shared vision with me, and so we developed it. So those are some of the reasons that I get involved in it. The most recent one is I was working with my wife's lab, because I wasn't able to get the microscopes that she had. So she had like 4 or 5 of these super resolution microscopes. And so I started working in the lab, and then I started working with the data, and I found I made enough of a contribution that I was a middle author, and I'm very proud of being a middle author. Yeah. So.
Yeah.
By the way, which book have you encoded in the DNA?
The one you have,
Regenesis.
Yeah, Regenesis. Yeah. It was actually a draft of it, but I included the photographs. So the photographs are also encoded in zeros and ones, JPEGs, compression algorithm, and so yes, I encoded that book. And in fact, then in the book, I wrote about the process of encoding the book in the book. So it was like very recursive in that respect.
Yeah, very Inception-like. Yeah.
Inception. Yeah, right.
So, in your interview with Stud, you shared how you live with necrolapsy and attributed your visionary ideas to necrolapsy. You said not as a bug, but as a feature. Although you still need to actively manage it, could you reflect on how you leverage this condition to be a strength?
Well, I think it maybe falls in the category of rationalization rather than leveraging, and maybe some mixture of the two. But you want to, if you could possibly do it, you want, especially if you're sort of have a positive view of life, you want to come up with a rationale. But so. I got a lot of chalk thrown at me as a student because professors took it personally when I fell asleep, like I'm wasting their time. I'm supposed to be taking notes. Maybe I stayed up all night playing video games, which I didn't, but I get excellent sleep at night. It's just not good enough. But yeah, very often I will fall asleep when I have a really tough problem. I just, can't figure it out, and I'll wake up with the answer. I mean, it's just, I don't know how to explain it. It's just there. Sometimes I'll have dreams while I'm going in and out, talking to people, say. It's not good, because I'll keep talking and I'm making less and less sense, and some people don't. I try to explain it to everybody so they don't take me, seriously when my eyes are drooping and I'm not making any sense. Because some people would say that I'm not making any sense even when I'm wide awake. But anyway, it's one of many sources of innovation that we can depend on. I forget the author who used to stick his feet in, maybe it was Shaw, in icy water to get new ideas. So there's all kinds of tricks.
Yeah, it's like everyone has their own ideas, and talking about the people as outliers too. It's like different kind of personalities or different routines that can help you get these ideas.
But this I had no choice on.
It just happened randomly, or does it have any trigger?
Well, there's genetics. So we've sequenced the genomes of my family, and There are narcolepsy genes floating around that are fairly rare. They're about one in, well, the disease itself is about 1 in 2,000 people. The alleles are a little more frequent than that, so there must be some combination, maybe a combination with the environment, maybe autoimmune. Yeah.
Cool. So we kind of wanted to talk a little bit more, like you're an entrepreneur and some of the science that you've commercialized. Sure. So you're one of the founders of Editas Medicine, which is seeking to develop CRISPR therapies for rare diseases. Since your lab was one of many that was working on repurposing these CRISPR-Cas9 systems to edit human cells, what was the process of seeing that this worked in the lab and to decide this should be commercialized since it could actually be used as a therapy?
Right. So part of it, we had already been working on lots of ways of editing genomes. Something that one of the first ones we worked on that probably most people haven't heard of is called SSAP, for single-stranded annealing proteins. But it works great in bacteria. It just doesn't work so well in mammals. And on zinc finger nucleases, I was part of a little company called Gendak that fused with Sangamo, which produced the first of the editing therapeutics to go into clinical trials, way before CRISPR was even discovered. And then Talons, and then CRISPR. And those last three were basically axes. They would cut the DNA. And we wanted something more subtle than that. Well, the SSAPs, the first of the set, was much more subtle. You could get precise edits. You could get homologous recombination. It was really what we wanted, but it wasn't. And it was even programmable with DNA, same way that CRISPR is programmable with RNA, which is its main attraction. It just didn't work in mammalian cells. So that's the luck of the draw. But we worked on all four of those, and others like recombinases, which are really exquisite things where they will rearrange DNA in big chunks, and that's really the holy grail, is to get that. But that does work in mammalian cells, but it's harder to program. So it's not RNA programmed or DNA programmed. So each of these 5 systems has its advantages, disadvantages. We had our second center of excellence. So I mentioned the first one, which was for next-gen sequencing. The second one turned out to be for next-gen editing. We were just going to be testing alleles that we got from the sequencing, but then we realized that zinc fingers wasn't as good as some other things we could do. And so almost all the people that started Editas were in some way involved. So, Keith Young was co-PI on the grant. Feng Zhang was a postdoc in the lab. David Liu was a close collaborator of all of us. And Jennifer Doudna wasn't part of that grant, but she had been a student in our department, and I reached out to her as soon as I realized that we were moving on from zinc fingers and talons to CRISPR, and wanted to make sure that she was, if she was going from biochemistry to human genetics, that we kept in touch. And so as soon as we all published things in January 2013, and then we all got together and started Editas. So that was part of the fun part, was really having all the pioneers on the same team. That's pretty rare that you get that. It didn't last that long, but it was pretty good while it did last. Yeah.
Yeah, and now we see with different companies using different CRISPR modalities like base editing or prime editing even to develop therapies. Yeah. And now that there's actually approved therapies.
And a lot of those people came out of Editas, or had co-founded Editas. They co-founded these other companies, like David Liu in particular, a fellow chemist and colleague at Harvard.
So there are obviously a lot of benefits to using CRISPR as a therapy, but there are some dangers that occur with some of the uses people are considering for it. Could you talk more about some of the ethical issues associated with it, since you've been vocal on the topics of bioethics?
Right, and this was another one of those ones, like DNA synthesis and personal genomics and gene drives. So one of the things you could do with CRISPR is gene drives, which is to spread things through wild populations to eliminate Lyme disease or malaria. And so it's a balance between the amazing good that could come from eliminating Lyme disease and malaria versus the very hypothetical that we could somehow hurt mosquitoes, or we could hurt things that eat mosquitoes or something like that. So, or the gene drives could spread to other species. Most of these have been mitigated, but But what we did was we wrote 3 papers on how to keep gene drives contained or reverse them first. That was our first salvo, rather than all the cool things and, let's get addicted to it first. Other things that came up with CRISPR were germline editing, which I felt was kind of a red herring, kind of a distraction. But it was amazing how it was like the train wrecks. You just can't look away from it, And in a way, I think that all the attention given to it, even though it had almost no purpose in life, there's almost everything you can do with germline, hypothetically do with germline, you can do with somatic, meaning conventional gene therapy, or even better yet, preventatively through genetic counseling, which is much cheaper than either germline or conventional gene therapy. Anyway, I think that all the brouhaha that we created about talking about it as if it were a real threat, created its own monster. It created J.K. Huh, who went to prison for 3 years for doing it for 2 or 3 children. And as new therapies go, it did pretty well. I mean, as far as I know, nobody got hurt yet, which is not true for a lot of other, like the first 2 gene therapies had both trials had people die. So it was both a red herring, both because nothing was going to go wrong in a FDA medical sense, but also because there was no use for it. So anyway, it had to play out, and it probably will continue to play out.
That's true. Just as these technologies become more kind of adoptable, there's always going to be some drawbacks or kind of issues that we haven't predicted might come up.
Yeah.
Yeah, and then you're also founder of the company Colossal Biosciences, which is seeking to de-extinct a few species of animals, including the woolly mammoth.
Right.
So what was this process like, from thinking of the idea for the company to working on it in your lab initially, and then securing the funding to start Colossal?
Yeah, so it's a very, as you said in the intro, I've started a few dozen companies, 50, and this was a very special one because it started basically as a response to a journalist, and that's part of the reason I like participating in podcasts and stuff. Is I find out what the public is interested in, and I get out of the box kind of questions, and maybe I think about them more than I should, and I thought, Nicholas Wade asked me for the New York Times. We had just, the world had just sequenced some minuscule amounts of mammoth DNA, and I think he correctly felt that was not a good New York Times story by itself, and so he started asking provocative questions like, well, if you could eventually sequence the whole genome, could you synthesize it? And I started sharpened up my pencil and figured out how much it would cost, which was a great underestimate, unfortunately. But after the interview was over and I thought about it some more, I said, yeah, this would be pretty good. But I didn't think it was that anybody would fund it. I didn't think the government, I didn't think philanthropy or industry would fund it. And so we kind of kept it on life support with spare change we had from royalties, and eventually philanthropy that I didn't expect came in through Peter Thiel. And I thought he was going to fund us for age-related diseases, but instead he picked this, and that kept us going again on life support, barely functioning relative to the other projects in the lab. It was about 1% of the research in the lab because that's about how much money we had. And then Ben Lamb came along, so Ben and I co-founded Colossal. But before we did, he just said, you guys really should have more money than you have, more than $10,000 a year. How about if I go out and raise $2 million? And I said, $10,000 to $2 million? Why not? If you can, I don't see how you can. And he did, and eventually, and then that was in 2021, and then as of 2024, 25, just 3 years later, he's raised $435 million, which is really, really remarkable on something I didn't think you could raise any at all. And that's completely changed what we can do. We can now look at endangered species in general. We can develop technologies. It's really a technology development company more than it is a de-extinction company. And what we're really doing is de-extincting DNA so we have diversity. One of the ironic critiques is that we're going to make less diversity. We're actually making more diversity because we can go back in time 2 million years and go all over the globe and combine DNA that normally the herds would never see each other, much less mate. And so I think it is a force of of diversity and also embracing your outliers. We're really looking for examples of there's some elephants that have no tusks, some elephants that have gigantic tusks, even bigger than anything, and elephants that are resistant to this herpes virus, and elephants that are extremely sensitive, and in fact, a lot of the extinction is going on. All the elephants are endangered species, but part of it is these herpes viruses.
Yeah, I think a lot of people are interested in it just because people think of Jurassic Park, and they're like, oh, they just think of that, and it's just such an exciting idea to think an animal that you normally see in movies or pictures, and you could see in real life.
Oh yeah,
and I guess
there's a great scene in the film where a paleontologist who's like carrying around a claw, a fossilized claw, suddenly sees the real thing, and you can just, as a scientist, you can just imagine what he's feeling. But there's an interesting link between, Michael Crichton was a student at Harvard Medical School, so I need to put that in there. But in addition, when I read his very first book on this in 1990, I saw a page of DNA, and you just don't see a whole page of DNA sequence in a novel, usually, at least in 1990. And so I wanted to know what it was, and we didn't have super good computational tools at the time, but I figured out that it actually was the DNA that I did in my rotation project as a first-year graduate student. It was PBR 322 that Greg Sutcliffe did for his thesis, and I helped a little bit during the rotation. And so it wasn't dinosaur DNA; it was recombinant bacterial DNA.
Okay. Oh.
So, and then one other connection was he came. They had this thing called a lysine contingency. They made all the dinosaurs need lysine in order to survive, and they would only provide that in their food at the park. So if they escaped, but what it missed was the thing that all medical students should have learned at Harvard Medical School, which is that all living things have lysine. So if you eat any living thing, plant, bacteria, animal, yeast, they're all going to give you lysine. So that was not a very good contingency. So since then, we have made a new amino acid that's only made chemically, not available in any living organism, and that does achieve what they wanted to do in biocontainment, which we consider an important thing to do.
Cool. Well, thank you so much for telling us more. I think the excitement of bringing back these animals is great, but also like the implications, because a lot of it's reintroducing these organisms and the climate impact too, hopefully positively impact the climate as well.
Right. Yes, that's one of the things that single out the species, or these species of closely related elephants. They probably interbred. If you look in their genome, there's lots of hybridization, just like there's hybridization between humans and Neanderthals and so forth. But the attraction of I would call it the Zimov theory, that the Arctic is really sequestering so much carbon, and if it melts, it could be released as methane, 1,400 gigatons, which is 100 times more than a human consumption of and production of carbon dioxide. So that's very tantalizing. Yes.
Cool. So what we want to talk about next is, in addition to you being the founder of several companies, trainees in your lab are also co-founders with you. So what has it been like to mentor trainees in the art of doing science, but also in entrepreneurship as well?
Yeah, so that wasn't something that happened right away. It took a while to evolve in the lab. I mean, partly because I was inexperienced both at running a lab and at running entrepreneurship. when I started. Obviously, I was mostly at the bench or at the computer. But with time, I think they would look at successes that I had starting things a different way, which was, I would get together with another faculty member or with a venture capitalist, and we'd start a company, which didn't involve anybody from my lab other than me. And they would look at that and they'd say, oh, that didn't look too hard. And then one of them, Francois Vignon, and another one, Luhan Yang, two of my student postdocs, said, well, why don't we become CEOs and or CSOs? And they did, and they were successful. And then all the other postdocs for every generation after that looked at those two and said, they made it look easy, and it's become one of the main exit strategies for leaving the lab is that when I first started, everybody would imagine that they would be a professor, or maybe they would work for some big pharma. But now none of them want to work. The professor doesn't get enough science funding money, not salary, but funding for the lab, and in big industry you don't have enough freedom.
Mm-hmm.
So having your own startup has the best of both worlds, academic and industrial, and you get the freedom and the money to because some of these projects really require $100 million to get even close to a clinical trial.
Mm-hmm. Yeah. It might speak to also the students and trainees in Boston too. I feel like it really draws people with the entrepreneurship mindset, and probably those are the students or trainees that really want to join your lab, and they kind of know in advance they want to start a company.
Yeah. So it's kind of Boston is a beacon for biotech, even more than I think the West Coast, where there's so many different beacons. Silicon Valley is mostly about silicon, not about medicine. So there's that beacon, and there's the beacon of Harvard and MIT, and then finally there's my own lab track record, which wasn't great to start out with, but it's pretty good now, and so they come with those expectations, and they help each other, and to my great pleasure, most of the alumni are very nice people, and they help new people enter the field rather than the kind of Hollywood stereotype of cutthroat corporate, sharks.
Yeah. So what are you, follow-up question, what are your bold suggestions for next generation entrepreneurs with the scientific backgrounds, especially, as you mentioned, the students, postdoc working in the lab right now who have idea of creating the startups?
Yeah, I don't feel like it's changing that much. I think we're getting slightly better at it. A lot of what we do, one motto that we haven't mentioned yet in this is failure is an option. So the NASA motto was failure is not an option. And of course, people and lots of money was at stake, and so that makes sense as their motto. But in our lab, the kind of the metaphor is that you have libraries of molecules, libraries of DNA and proteins, and you go through. The library might have millions or billions of components in it, and you're going through for that needle in the haystack. So you really expect millions of failures in order to get one or two successes. And so ours is much more of a failure culture, if you will. So that's one thing. And so now, of course, when you start up, you don't want, and I think we have a fairly low rate of failure in a startup, but the ideas that go into it, you want to kind of quickly whip through them. And I think that kind of attitude of setting up libraries of ideas, libraries of molecules, it permeates through a lot of the startups.
The next question is the commercialization of longevity research has long been a controversial field full of buzz, with many claims but a few clinically validated results. So in the recent case and in the Netflix show Don't Die, businessman Brian Johnson shares his conventional anti-aging lifestyle. He claims his method is backed up by science, though most of this science is still in the very early clinical stage. You co-founded Rejuvenate Bio, a biotech company focused on developing gene therapy to age-related disease in both humans and animals. So what's your vision for humans' longevity research commercialization, and what do you believe or not believe?
Right. So I think, aging reversal or even solving age-related diseases got tangled up with longevity and immortality, which is extremely difficult to prove to the FDA that you have an immortality drug, because that would take an infinite clinical trial. And even longevity would take a 30-year clinical trial, which is unaffordable. So that was one complication. The other was that there was this hope that it would be something simple, like something you eat or don't drink or don't drink. Ideally something that's natural, so you don't even need FDA approval. And that resulted in just a lot of wishful thinking. And I don't think everybody was a charlatan at that point. This is before I entered the field. I think that they just convinced themselves, or they wanted to believe it. And you did have a lot of anecdotes. You'd have people that were self-medicating, but not doing randomized clinical trial, which is what you really need. And so a lot of these people would consult with me as I entered the field, and I started publishing papers. Thanks to Pedro de Magalis, one of my postdocs, got me started in this field in the early 90s. They would ask me, and I said, look, if you want to be a guinea pig, and I've been a guinea pig in many research studies, do it in a randomized clinical trial, But nobody wants to be the control, right? They all want to live longer, and they want to start it right away. And I don't think there's that big a rush. But anyway, even if you don't want to have any controls, or even if you don't want to be the control, you can do a crossover study, which is basically everybody at some point is the control and is the experimental, and you don't know which it is. Anyway, so people like Ray Kurzweil and Brian Johnson and others, set themselves up as role models, even though they're spending like $1 million, $2 million a year, which is not really affordable, and that's part of ethics. It's the simple ethics of the FDA is, do no harm, so that it's safe and effective. But there's another ethics, which is affordable, and so that everybody can use it. But moreover, it's all biased towards this. Oh, it's something simple you can eat or drink. Some pill that's already been approved. And I think aging is a much more complicated disease. Complicated doesn't mean that it's far off. It doesn't mean that it's a century off. It could, be the one more cycle of FDA approval away. But it is. We need to respect the complexity of, and we know a great deal. We may even know more than enough to do the engineering. And so Rejuvenate Bio that you mentioned came out of a postdoc in my lab, Noah Davidson, who's still, was co-founder, still there, working on it, and has done it very, very low cost, and has 3 clinical trials now going on, done the right way, not self-experimentation, but done on both dogs, which is a veterinary product, and in humans, which is a standard human clinical trial. And I think that's the way you should do it. There are also bigger companies that are not any further ahead, but still, I think, contributing to the feeling that we have a real field now rather than charlatans, which is Altos Labs and before them Calico. And so these 3 companies and a few others are actually getting things into clinical trials that look very promising, and they're not simple things that have already been approved. They're really a lot of research. Probably $6 billion went into 2 of those companies, and maybe a few million went into our scrappy little company. But I think they're all. I can, really interested in what comes there. it's transformative not just because nobody wants to die. I think that's kind of sophomoric. It's really because almost every disease is affected by aging, and 90% of us are going to die of it. So if we actually care about medicine, which is our mandate at Harvard Medical School, we should be paying attention to that as much or more than anything else.
That's really promising to hear. I feel like people kind of brush off aging just because they haven't seen results for decades of work, but it's good to hear with Altos, Rejuvenate, and what Calica's doing. There's like promise.
Yeah.
And then we wanted to ask, a number of biotech startups are focused on AI-driven approaches, particularly for protein design and drug development.
Yeah.
So what do you think, how do you think AI will impact biotech research in the near future?
Great question. In fact, I will take the bold position that the most interesting thing so far from AI is protein design, not protein three-dimensional structure, not ChatGPT, not making artistic things. Although art is so broad that it's fairly easy to succeed, at least succeed temporarily, until the rest of the art world wakes up. So I think protein design is really working. The large language models, they allow you to make a great diversity of proteins that all work. It used to be when you made that kind of diversity by random mutations, you'd break the protein or you'd break the system. But now you can make almost as many changes as you want and still have a functional protein, and that increases the chances it has a new function. And you can even design it towards those new functions. And this is just going very, very quickly. Our first paper on it was in 2019, and we now have 5-6 companies that are exploiting that intersection. It's actually a three-way intersection, not just AI and protein design, but the third component is these big libraries like this that inspire us to fail a lot. If you design the libraries using AI, then they're going to fail less, and that means you can push them harder, and you can test them in a way that would previously been uneconomical. So we tested 1 million different viral capsids for gene therapy deliveries in primates. Now, if that involved 1 million primate trials, nobody could have afforded that. Even big pharma couldn't afford that. But we did all 1 million tests in one primate, and then repeated it in other species of primates.
That's a really good point. I think we mentioned earlier about the failing a lot, failing fast, and AI kind of helps you to do that without the waste and like. Well,
you throw out a bunch of things computationally, and then you still have 1 million left over, and then you throw out 99.9% of those, and then what's left is really worth the deep dive in the clinical trials.
Yeah, no, that's like a perfect way to word it.
Yeah.
And then we kind of want to learn more about people, these scientists, but outside of science as well. So we're wondering, could you tell us a little bit more about your hobbies? What do you do outside of research?
I had tons of hobbies when I was young. I would say for the last 30 years or so, I've been following the hobbies of my daughter and her 2 daughters, so my 2 granddaughters, because it's fascinating. It's a chance to learn new things, things that I wasn't already doing. But I've done vertical rock climbing and cliff diving and all that kind of stuff when I was young, and now I'm interested in art, photography, fashion, so forth, because I see things through their eyes, and it's a blessing.
Yeah, it's really cool to hear adopt new hobbies as your family grows, and and then. We're in your office right now. We see a ton of books on there. Could you tell us a little bit more about what are your favorite books?
It doesn't look like the hobby of a dyslexic. I didn't overcome my narcolepsy, but I did mostly overcome my dyslexia. With your brain is constantly reprogramming. It's just an amazing thing. I watched a member of the family that had a stroke, and she had lost her ability to walk or talk. And then just a few months later, she's, you could barely tell it. It's just amazing. So, yeah, these books are mostly either science books or written by friends, and they send me the books. Some of them are novels that have to do with science. Some of them involved people interviewed me briefly and were for either the nonfiction or the fiction. So there's a little story behind each of them. Yeah. I don't know if that was your question.
Oh, yeah. Hearing more about the books and if there's like a particular book that's your favorite, or what are some of your favorite books?
Definitely. I think people should read Genesis.
Read Genesis by George Church.
Yeah, right. Just in case I'm grabbing one of their copies of the book. Mine is buried somewhere, but no, that's highly biased. I have a big stack of Walter Isaacson's books because he interviewed me and he was kind enough to give me a signed copy of each of his previous books. I like. Yeah, there's a whole variety of things that I like that are kind of quirky and weird. I don't know. I don't even know where to start. I pull them off from time to time, but these days more and more stuff comes from the fire hydrant of the internet. Where you can go through PubMed articles, and very often I'm not reading the whole thing. Sometimes I do, sometimes no. But the point is, you could read bits of 10 papers in a few seconds, and that's complementary to how we used to do things, where I would have to go across town to the Harvard Medical School library and photocopy for a while and take them back and read them cover to cover. That's not the only way to do it.
Yeah, it's much faster now. Social media, people.
Yeah, people will bring it to my attention, and there's some pretty good filters. It's also possible to put yourself in a little echo chamber where you're only listening to falsehoods. But I'm fully addicted to finding the difference between things that, and scientists are perfectly capable of self-deception for short periods of time. But it's wonderful to see how it self-corrects.
Yep. Yeah. Do you have my favorite movies or some movie you like?
Well, we mentioned one, the Jurassic Park series. What else? For some reason, I love Monk. There's a TV series about this guy that he's a neuroatypical.
He's a germaphobe, right?
Well, I think OCD is probably the diagnosis. It's not just germs, and it's just, and he solves problems that other people can't solve, and they kind of set up they set up the solution so that you sort of feel like you could solve it too. Yeah. So that's an interesting one. There's no shortage. A lot of them are films or books of my friends that I look at to see, the films based on books. I think it's it's easy to dismiss it as something where you're being too passive. There's this worry about people are spending too much time looking at their phones. But before that, there was a worry that people were looking at their books too much, and even in Gutenberg's time, it was a worry that people that were not properly educated were getting access to books, that weren't part of the clergy, and so there's always been this kind of thing. But I think it's movies are fairly they can be a way that you can quickly and deeply get engaged in a topic, much more so than a book. Books will, sometimes I'll read a book after I've seen the movie, because it expands a 2-hour experience into a 15-hour experience, and there's a lot more detail there, and you can take notes and look things up and things like that.
Yeah, and people that are like visual learners too, they get a lot more from seeing movies, TV shows.
Visual, emotional, things that are, I think the emotions that I feel, if you're a really spectacular writer, you can get some of the same emotions, but boy, it's usually more powerful with the visuals. Yeah.
Cool. Well, with Fung and I agree that this was like a great conversation going to science, scientific journey, mentorship, entrepreneurship, and learning a little bit more about yourself.
Yeah.
So we really appreciate you taking the time to
Thank you so much.
be in the interview,
Yeah.
and also being a big supporter of our club, and also trainees at Harvard for about 40 years now for your lab, right?
Yeah.
So yeah, we really appreciate it, and I think a lot of people, our listeners, are going to really like to hear about what we talked about as well, since these were all things that we thought were really interesting.
Okay. That's why I'm here. I mean, here in this podcast. It's not why I'm on Earth necessarily, but yeah, it's lovely. Yeah, I'm very interested in what people want to talk about. Yeah.
Cool. Well, thank you, George.
Yeah.
Yep.
Thank you, George.
Hi, everyone. That's the end of this episode for the Harvard Biotech Club's The Pulse podcast. I want to thank Fung for helping co-host and produce this episode, and I want to thank everyone for listening, and please stay tuned for our next episode with Sufyan Abulhuda, CEO and co-founder of Nucleate.